I put here.
Great. Okay. So it's great to be here. Thank you, everyone, for staying through to the bitter end today, and thank you for organising this conference, to Mario and to everybody else. So I'm going to skip the first slide because we all know about this now. I'm very lucky because so much of the background has already been explained that I don't have to repeat it. For example, this slide was shown before, and I was going to show this to explain why the issue of opioid use disorder is quite so problematic, and of course it's related to the extent of the epidemic. It's a national health emergency in the United States. The problem is coming here to a much greater extent than it was. The data show that, overdose rates are extremely high. So the focus today is going to be on a couple of things, which is what are the long-term mechanisms that underlie the development and the long-term nature of opioid use studied in rats, and also what might some of the epigenetic mechanisms be underlying individual differences in vulnerability to disorder. In the meantime, I'm stuck with these two gentlemen, but next time I'm going to use my own family and find a little way an excuse to include them instead. So, but this is just to illustrate the idea of how people's lives can take very dramatically different paths, even when they appear to come from relatively similar backgrounds. Some of you might have heard of Wes Moore. He's currently the governor of Maryland. He's a possible presidential candidate, former Rhodes Scholar, which some of you will know about, and he was a business leader, the Robin Hood Foundation he founded, a pretty amazing guy. And he wrote a book called The Other Wes Moore, because he discovered that there was someone else with the same name as him, by the name of Wes Moore, who lived a block away from him. And whereas he became this Rhodes Scholar, business leader, governor of Maryland, he wrote before some of those things had taken place, the other Wes Moore entered a life of drug addiction, gang violence, and he's currently serving a life in prison. And obviously there are quite a few differences. He spoke about the family and the influence of his mother, etc. But there is still a question about why do these paths diverge? And why does one person go one way and another go another? And this is something that we've also heard already from Shaul today, which is why is this important? It's important medically and financially because if we can understand individual differences in vulnerability and individual differences in trajectory, we can then tailor things to those people's needs. So now I'm going to get to what I'm going to talk about. This was from a review paper that we once wrote. And essentially what we're interested in is, if I can use the points, is it the red? Yeah. Individual differences in vulnerability to substance use disorder, specifically it's going to be opioid use disorder, that produce in some people long-term substance use habits and in others don't. And as probably all of you are aware, especially after you've heard all the talks today, there are logically two only possible factors, what Zaki called destiny or genetic variation, the variations in bases between individuals, and environmental influences. And so these can have their own effects, they can interact, and then there's also a residual effect. So where do these things come together, and how does that, and also what underlies the change in behaviors that produce to these dramatic long-term consequences or sequelae? And the answer here is at the level of epigenetic regulation. It really has to be the case. So what does epigenetic regulation mean? Some people think it's all to do with non-genomic inheritance. That's only a very small part of the story. It's the long-term cellular changes in the way in which cells are regulated. So it's important in anything. It could be memory, it could be height, it could be aging, it could be anything. And we're going to talk about drug addiction. And both genetic variation and environmental influence and experience with drugs, which is a kind of environmental influence, all going to feed into that epigenetic regulation. What I'm going to show you today in a rat model is we're going to look at long-term substance use, opioid use in rats, but we're going to look across the trajectory, so from early to late, and look at differences and similarities in the epigenetic mechanisms involved, I'm also going to do this brief exposure, which is an early model of addiction. And we're going to measure gene activity using RNA-seq. I'll show you that in a minute. Epigenetic regulation. I'm going to show you how we do that in a minute. And also we're going to relate it to genetic variation by looking at the results of other very large studies. You need large numbers of subject rats or humans to look at genetic variation. But then you can integrate your data with genetic variation, which we did here. So you've seen this before. The gold standard for looking at voluntary drug taking that's somewhat similar to a human voluntary drug or volitional drug taking is the self-administration paradigm. Morphine self-administration in this case. And in this large study, we looked at 3 different behavioral paradigms to look at what we can call early indicator of OUD, persistent or chronic, or relapsing. Remember, we had a slide earlier saying that the definition of substance use disorders is that they're chronic and relapsing. So we wanted a measure of each of these. So for early, this is supposed to be someone experiencing anhedonia. Negative affective signs occur right from the first time individuals take drugs during withdrawal. So we're going to look at withdrawal-induced anhedonia. They only get larger over time, but they're the most consistent sign of opioid dependence from the beginning to the end. So that's going to be our early measure. For persistence, this shows you a sale. Okay, it could now be on Amazon, but people's behavior is very influenced by the price of things. And a measure of chronic drug addiction or chronic anything or persistence is how long people will continue to behave in a certain manner as the price goes up. That's a behavioral economic measure, and we use what's called demand elasticity or alpha, which is very similar to the progressive ratio that somebody showed earlier, except that it's one thing that we test on a day over a longer period of days, so it's slightly different. And finally, for relapse, with opioid addiction, relapse rates are very high. We saw a slide earlier again. It's great I don't have to show you any of this stuff. Up to about 80%. We're going to use the classic measure of extinction followed by reinstatement, where you give some cue to remind the person, or in this case the animal, of what they're missing, and then they start to respond again. And we use both morphine and a cue that was associated with morphine for this. Okay. One other advantage to that early measure of withdrawal-induced anhedonia, it's not only that it's an early measure of opioid use disorder dependence, but also, somewhat fortuitously, it correlates, we found, with subsequent measures of morphine self-administration. So animals that showed high levels of spontaneous withdrawal. Spontaneous means we don't, instead of giving naloxone, we just let the drug wear off. So a few hours later, they'll start to show anhedonia. The more anhedonia they show over the first week, let's say, the lower these are. So it's negatively correlated, but it's a predictive measure, and there aren't that many predictive measures, but we've actually also heard about some of those today, like how craving can be a predictive measure for later vulnerability to substance use. So this just shows you the behaviors. This is kind of not the most interesting part, but this shows you how, I'm not going to explain ICSS thresholds unless anyone wants me to, I can later. It's elevated thresholds in intracranial self-stimulation as a measure of anhedonia, which is quite tricky to do. But you can see how that increases over days of giving morphine. The animals aren't yet self-administering. It goes up over days as you get more and more anhedonia 23 hours later during withdrawal. We also see very robust reinstatement. And this is a demand curve from which you can measure alpha. And it shows us the unit price of the drug, which is given for those who know about these things by the FR ratio. As the FR ratio goes up, the amount that the animals take goes down, and you can have different curves for this, and you can calculate from that your alpha or your elasticity of demand. One thing which is the curse of behavioral, animal scientists is usually when you get a lot of variability. Then I need a bigger N. But here, variability is a strength, And we do see variability here because what we're going to want to do is to look at animals across the spectrum of vulnerability. So we want to be able to split them into groups that show high or low variability. Okay, before I get to the data, I'll just show you what we did. We had kind of fairly large groups for this. We had these 3 different paradigms, and for each one we would develop a behavioral score 24 hours after the end of the experiment, which is a significant factor. I can explain later if anyone wants to know. We dissected the brain, and we specifically looked at the medial prefrontal cortex. The great thing about this audience is everyone's been talking about the medial prefrontal cortex. Sometimes people say, why the medial prefrontal cortex, not the accumbens? So you all know why. It's got great translational value, etc. And then we looked at 3 different things, or one we're looking at now. So one is gene expression. So you can see here that for this, the DNA is spooled around your large histone molecules. Together they're called chromatin. And you can look at gene expression here of a target gene. The other thing that you can do, many things you can do, and you're going to look across the entire genome. So it's an unbiased approach. You look at about 22,000 genes. You can also, look at how open the chromatin is using something called transposon binding. So basically, when it's tightly wound like this, genes aren't expressed. But the mechanism of gene expression is that the chromatin becomes more relaxed through various processes, and that provides access to these TFs, or transcription factors, to bind to either enhancer regions or closer to the gene promoter regions and encourage the gene production. So we can look at where these open areas are, and we can identify which transcription factors bind to each. The third thing that I'm not going to talk about today that we're doing now, and we're going to be doing more of, is we're going to look at DNA methylation, which we just heard about, and also gene sequencing. So this is methylation of the chromatin, which is something different. The DNA itself is either methylated or something called hydroxymethylated. And with this new technology, we can actually look at methylation and hydroxymethylation across the genome. And we can also sequence to an individual base level. So we can look at the whole genome sequencing, but again, too early for me to show you that. Okay, so this just shows you the long-read sequencing I'm not going to talk about, but we had RNA-seq and we had ATAC-seq. So what I have in the time remaining to me is about 8 findings that I'm just going to go through very, very quickly. They're kind of some big take-home messages from the study, so don't worry about the details. And actually, unfortunately, the first one is probably the most opaque to follow, so I shouldn't have started, but I wanted to get the ATAC-seq out of the way quickly. So remember, the ATAC-seq is a measure of this open chromatin, and then using something called HOMA, you can actually identify which sequences here, binding motifs, are particularly enriched, and that they are associated with specific transcription factors. So these are transcription factors. Strictly speaking, though, that it's not really just FOS. It's a family of, FOS is part of a family of about 20. Someone here might know the exact number. MEF2, there's 2A, 2B, 2C, 2D. So there's plenty of these. Also, even PGR, progesterone receptor, could also be glucocorticoid receptor, which is very interesting. But the take-home with this particular slide is that if you compare morphine versus saline animals, you get one set of findings. That's what most people do in their studies, right? They say, these animals got the drug, these didn't. But if you now compare resilient and vulnerable across the 3 paradigms, you get different patterns, okay? So there's some aspects of gene transcription that are a function of simply an animal getting morphine or becoming addicted to morphine, but there's other factors that are more related to individual differences. And if you don't look at individual differences, you're not going to see that latter set of factors, okay? The second point, which is probably, I think, the most interesting of everything that I'm going to show you, to me, I'm biasing you already, is the following. So we can look at what are called canonical pathways. So you look at all the downstream genes that are differentially expressed across the 3 paradigms, And yeah, you'll see immune stuff up there. And then what you can do is use a gene ontology tool to figure out which canonical pathways, that's like which functional, molecular, they could be biological pathways, cellular pathways, or whatever, it depends on how you search, are significantly either up or down regulated. Okay? And what you'll see here, which is very striking, if we compare resilient, not resilient versus vulnerable anymore, that doesn't give you this. You have to look at resilient saline, the resilience against the controls, or the vulnerable against the control. Where do you see most of the activated or inactivated pathways? It's in the resilient animals, less so in demand. By the way, one thing I didn't mention here is sex. That's a whole other factor, huge thing with genomics. This was true in males and females. What that means is the resilient animals are the ones who show most of the changes. And by the way, this just shows you some of the pathways. Particularly strongly changed pathways were immune function and plasticity. But that means if you're doing a study where like a biological study, and you find some gene is affected or some pathway is changed, our immediate assumption is we need to knock that down. But actually it might be you want to build it up, right? So that's another significant finding. And in fact, directionality is a whole other story. That's a whole other thing. Is it blue or orange? That's determined a lot by when you sacrifice the animals. That's a huge issue too. So this just down below shows you how you do RNA-seq. You can read that in your own time. Okay, our third kind of lesson here is that this resilience vulnerability phenotype, whether animals are vulnerable or resilient, remember, I'm looking across three totally different paradigms. Animals get a little drug, they get a lot of drug, it can be months, it can be days. Regardless, you can look at what's the contribution of that phenotype to genetic variability versus what is the contribution of the paradigm. And to do that, we use a technique called variance partitioning analysis. It parses out those two factors. And you can see the largest, not surprisingly, is paradigm. That's not surprising at all, right? Because they're so different. But there is also quite a lot of variability associated with vulnerability across the three paradigms. This is very hard to read, don't worry about it. This shows you the top 50 genes where their variance was most attributable to the vulnerability, resilience, not to the paradigm. And the important thing is that when you do the gene ontology analysis, it's epigenetic mechanisms. So it suggests if there's a core trait of vulnerability to drug abuse, it's related regardless of time, amount of drug, what your paradigm is, independent of that, it's related to epigenetic processes. Okay, if I haven't lost you, our fourth one, we see some things that are specific to a particular phase. And I think the most interesting one is oligodendrocytes. So everyone thinks about neurons. Nowadays people are thinking more about astrocytes, Peter Kalivas, come back to him a bit later. Some people are thinking about microglia, someone had a slide on that earlier, but fewer about oligodendrocytes. But what we found, this shows you what an oligodendrocyte is, okay, that's not my data. These are the cells that provide the myelin sheath for neurons in the brain and affect white matter pathways, neural conduction, etc. And what we found through another technique called a weighted gene co-expression network analysis, or WGCNA, was a module of 120 genes, of which 29 genes were specifically related to oligodendrogenesis, which is an extraordinarily high number. So what that means is that with this bulk DNA sequencing, we've detected that there's this factor associated with vulnerability, which is associated with oligodendrocyte development. And what I was surprised to find, because I didn't know anything about oligodendrocytes, is in our brains we've got a ton of these oligodendrocyte precursor cells. How many people knew about these cells? Maybe a few of you, right? And they're just sitting there. You think your brain plasticity, your development is done by the age of 6. They're sitting there throughout your lifetime. They can be activated at any time, and that changes conductivity. It happens when you're injured. And people who have addiction, there are differences in white matter. There's a lot of evidence of that. So this looks like it's something. So this is one of the things we're interested in. This just shows you the pathways, right? The gene ontology analysis. So this is one thing we're interested in targeting. Okay, moving on to our next one. This is related to a collaboration we're doing with A. Palmer's group at UCSD. So very, very great foresight. NIDA, about 30 years ago, started a colony of rats called heterogeneous stock rats. So, most of the rats we use Sprague-Dawley usually. Sprague-Dawley? Okay. They use mice, even worse, because they're all clones, okay? But even Sprague-Dawley rats, what they have found is they don't show a massive amount of genetic variability. So what they did was they took 8 founder lines of rats from different lines, put them together. Now, in his lab, Palmer's lab, I was at the 100th birthday. That means that they had a cake for the 100th generation of rats that have been generated. The advantage of this is you've got massive genetic diversity, it's much more like humans, and very small what are called haplotypes. Smaller haplotypes means that you can hone in much more accurately with smaller numbers of subjects into genetic variants that are different between individuals. So one of the studies they've done on this, so they've got huge numbers of animals that my colleague over there, Olivier George, has studied, where they've done a very long drug self-administration with either cocaine or oxycodone. So using the opioid data, so this is actually another study. So this was by Brittany Kuhn and Peter Kalivas. They were looking at heroin. They looked at several hundred HS rats, and they came out, you'll see how this is relevant to my data in a minute, with this kind of, you may have seen, a porcupine or Manhattan plot, and this shows you some interesting variants, right? Where there's some genetic variant, this is chromosome 11 over here, this variant over here, we don't know exactly which gene it's in, but we can kind of approximate, is related to the level of consumption of heroin. Okay. So they found a number of variants. If you zoom in on that particular variant, this is the region of the chromosome. So these are the candidate genes and regulatory regions that are important. One thing that they reported as well, they've done something called an EQTL analysis, an expression quantitative trait loci analysis, and that maps on variants to gene expression within a certain area of the brain or body. What they found is that in the same region there were EQTLs for expression of 2 genes, SH3BGR and PCP4, in the medial prefrontal cortex, which is the area of the brain. So now we've got 2 pieces of very interesting information. We know there's a genetic variant in this line of rats, which is not our line, so we don't know if it's going to work in ours, that's important in vulnerability or how much drug animals take. And we know that there's 2 genes in that area whose expression seems to be affected by a variant in that area. So the question we asked was, well, how much does drug taking correlate with gene expression in the prefrontal cortex in our study? And what we found was that with reverse directionality in demand and in reinstatement, our 2 long-term measures, there was a very significant correlation. Remember, this is the first a priori data I'm showing you for PCP4, not for the other gene. This is the only a priori thing that we did. So we only looked at 2 genes. To find a high correlation is pretty surprising, right? And just in July, a paper, no one really knows what it does. It's called Purkinje cell protein 4. So it's something to do with the cerebellum, but it turns out it's really important elsewhere in the brain. I can explain why later. 5 minutes left. And basically, it's important in dendritic growth and maintenance of dendrites, which is obviously very important in plasticity. So a really interesting gene that we want to follow up on. Okay. What this just shows you that there's a high overlap between what we see and drug addiction data in individuals who died of opioid use disorder through overdose. So that just shows some translational significance. There's 2 more findings I want to show you. The first one is an analysis across multiple data sets with our colleague Jia Yang. So Jia Yang, UCLA, analyzes multiple genomics data sets and then finds what we call key drivers, or genes that can account for variations in gene activity through all these different metrics. So we plugged into her system our ATAC-seq data, our RNA-seq data, those two things I showed you, and that GWAS data from the Brittany Kuhn study, from these completely different animals. Came up with this very complicated network you don't have to worry about, but the dopamine, not surprisingly, receptors came out from this as highly likely key drivers. But there's one other thing I want to say, and this is what I'm going to finish with, is she did an additional analysis that she calls pharmomics. So what she now does is she maps all of what I just showed you to the profiles, the fingerprints, or the footprints, whatever you want to call them, of the profiles of drugs, drug effects on gene expression. There's a huge database out there. Drug X affects 46 genes. Drug Y. So then what you can do is try and match the drug to the pattern that we see, and that allows you to maybe repurpose drugs that we know already exist and may be very cheap. Okay, so this is the top 10 drugs that came out. Some of them useless, they would kill people, they're for very serious cancers. Some you can see, dexamethasone is up there. We're not going to use that for addiction, but it shows the relevance of glucocorticoids. And COX-2 inhibitors, people are studying that. The last thing I want to show you is this. Two drugs came up, one metformin, the other with a name that I can't pronounce. And these two drugs are used either individually or in combination to treat type 2 diabetes. What does that sound familiar to? GLP-1 inhibitors, right? So, of course, the GLP inhibitors aren't on this list because it's too recent. But so our question is, maybe these drugs are very important in addiction. So the very final data slide I'm going to show you shows that indeed we find in males and females that when we give metformin, we block opioid self-administration. A similar thing has been found by my former colleague Sade Spencer, who some people know here, with cocaine. She's been studying metformin for a few years, and she's doing some really interesting stuff on it too. So this is another interesting direction for us. I'm not going to bother sharing with you the final story. It's someone Abby and I met, who was when he told him about my work, this was in the Delta Lounge in LAX, his incredible story of recovery from addiction. If anyone wants to hear it, I'll show it another time. I'll finish. I just want to particularly thank Shirelle, who was my graduate student, did a lot of this, and Andy Harris, who has been my partner in crime throughout this. Thank you very much.